Okay, now we're going to be going over Chapter 4. In this chapter, we're talking about cells and all of the different organelles and their functions within the cell. So, as you guys recall, we mentioned this before. When we're talking about the cell theory, this is the theory that states the importance of cells to life. So, the three main points are that all organisms are composed of one or more cells. Cells are the smallest living things, and cells will arise only by division of previously existing cells. When we're looking at the size of cells and their contents, this picture is pretty helpful. You can see that we've got 20 millimeters here when we're looking at this section of a thumbprint. As we zoom in even further, we're down to 2 millimeters. 0.2 millimeters is including one cell along with some other adjacent pieces of other cells. Now we're down to 20 micrometers. You can see the definition within the cell. We've got different organelles in here. You can see our nucleus and the other organelles that we're about to introduce. This is one specific organelle called the mitochondria. As we zoom in even further, down to 0.2 micrometers, you can see that we are within the mitochondria. We're looking at specific proteins. And then we get all the way down to 2 nanometers, and we're looking at the specific atoms that are bound together into molecules to create these organelles. So most of our cells are going to be very small, and there's a reason for that. The first cells that were actually observed under a microscope were discovered by Robert Hooke in 1665, which is pretty amazing to think that he was able to accomplish that so early in science. We also have recognition from Matthias Schleiden in 1838, who was the first person to recognize that cells were a fundamental part to plant composition specifically. And then about a year later, Theodor Schwann reported that all animal tissues are also comprised of cells. So you can see we've got a long distance between the first description of a cell using a microscope in 1665 to the realization that all plants and animals are composed of cells, which was roughly 200 years later. Cells are not all the same size, but most cells are going to be small, and that's because it just makes more sense. Larger cells are not going to function as efficiently. And that's because they're more difficult to control, which is because of the distance between the command center at the core, which is the nucleus, and all of the other surrounding regions. Organisms that are comprised of many small cells are at an efficiency advantage, so they're able to do things more quickly over organisms comprised of few larger cells. One way we could think of this of when we're trying to make sense of why large cells are just not as efficient. You could imagine if you're trying to deliver a message to a specific area within a building. If you have to walk into Frank Hall and that's all the information you have, and you have to find a specific table to set this file folder down onto, you can imagine that's going to take a lot longer than if you were just given the task of going straight in to the first office on the right on the first floor and delivering that message. So you could see if we've got a smaller distance to travel to our destination, we're going to have a more efficient way of completing all of our processes. And that's really all we're talking about with this surface area to volume ratio. If we have a smaller surface area, meaning the surface area of this cell to the volume that is inside it, that's going to make it easier for materials to diffuse and find the exact spot they're trying to reach. Smaller cells will have a greater surface area compared to their volume, and as cell size increases, the volume is going to grow more rapidly than the surface area. Here you can see another example of our surface to volume ratio. You can see a smaller surface area here with a smaller volume. And here we've got a larger example of a cell, and it's easy to visualize when looking at how much faster we would be able to reach this nucleus if all of these small cells were adjacent to each other than it would be to make it all the way to this nucleus if there were a bunch of this size cells adjacent to each other. Sorry, forgive my voice. I have been sick, so it's not it's not ideal, but we're making it. When we're talking about resolution, it refers to the minimum distance that 2 points can be apart and still be distinguished as 2 separate points. The human eye's resolution is about 100 micrometers, but we have found ways to increase that resolution, which we are able to do through magnification, which is what we're really doing when we're using microscopes. We're increasing the magnification so that we have a better resolution. We have different types of microscopes that are used to increase magnification. One of the most commonly used is the compound light microscope, and this uses sets of magnifying lenses to resolve structures that are separated by more than 200 nanometers. When we look at electron microscopes, they have 1,000 times the resolving power of those light microscopes that we just mentioned, and they can resolve objects as close as 0.2 nanometers apart, which, if you'll remember from our previous slide here, that's 0.2 nanometers apart. So we're on the atomic level when we're using a scanning electron microscope. And here we're looking at the range that we're able to observe with a compound light microscope. So we're able to see from 1 centimeter all the way down to a little past 1 micrometer, which is our bacteria. So here's the range of our compound light microscope that you guys will be using in lab. And then here's the range of our scanning electron microscope. So you can see if we're getting down to the atomic level or molecular level, or we're trying to look at viruses, we're going to need this scanning electron microscope because we're going to need a higher resolving power. Here's some examples of what it might look like to look at various things through a compound light microscope. Up here on the top we can see 25 times resolution for onion cells. And then as we zoom in to 100 times and 1,000 times resolution, we're getting a clearer picture of those onion cells. Here you can see an image of human cheek cells. Here we're looking at an amoeba. This is the bacteria Streptococcus aureus. And then here is the bacteria Bacillus anthracis. Scanning electron microscopes are able to get to such a fine range that it almost looks computer generated whenever we are looking at these images. You can tell we're looking at Staphylococcus aureus again with a much better resolution than it was in the previous image, where this is all we could hope to see. Here are red blood cells. This is the face of a mosquito under a scanning electron microscope. And then here is an example of pollen. As we begin taking a look at the cell and all of its specific organelles, it makes sense to start at the outside. So when we're looking at a cell and we're looking at this outside ring around the cell, that's going to be made up of the cell membrane. And we've talked about this just a little bit. We've touched on the phospholipid bilayer, which you can see represented here. Here are phospholipids with their polar heads and nonpolar tails. And here are the various proteins that are scattered throughout that cell membrane as well. As we continue, you guys are going to see cell membrane and plasma membrane kind of used interchangeably. Those are the same thing, so there's no difference. If you're talking about the plasma membrane, that's the same as that outer membrane surrounding the cell, also known as the cell membrane. It's conceptualized by the fluid mosaic model, which we have mentioned before as well. This membrane is made up of a sheet of lipids with embedded proteins, and that lipid layer is going to form the foundation of the membrane. Then we're going to have different things scattered throughout, different kinds of proteins. You know, we've got our cholesterol in there to control the rigidity of our cell membrane. And the majority of it is made up of that lipid bilayer composed of those phospholipids. So when we mentioned the fluid mosaic model, I included this picture just so that we could keep in mind the only reason that we're including mosaic in this name is because we're trying to express that we're showing the individual pieces of our plasma membrane, of our cell membrane, with this model, with this image. We include the word fluid because none of these pieces that we see represented here are necessarily going to stay there. That's just where they are in this one representation. But all of the pieces in these membranes, this glycoprotein could easily move and end up on the other side over here. This cholesterol could end up over here. It is fluid. So none of these things are really locked in place. They're able to move around, and that's what makes them fluid. So the reason I included these images is to just show you, just like with a mosaic piece of art, we're able to see the individual pieces that make up the overall picture. And that's what our goal is with the fluid mosaic model when we're trying to represent that plasma membrane. We're just trying to show the individual pieces that make up the whole thing of our plasma membrane. Here you can see a zoomed in view of a single one of our phospholipids that's making up our phospholipid bilayer. Here you can see our polar head to that and our nonpolar tail. So we have these two nonpolar tails attached to a polar head. And the polar region is what contains a phosphate chemical group, and the polarity is what allows it to be water soluble. So this region is water soluble. But the nonpolar region is comprised of fatty acids, and therefore it is water insoluble. Whenever I say water insoluble, that's just a way of saying not dissolvable in water, which makes sense because that's our nonpolar lipid, just like how we talked about oil being a nonpolar lipid, and the reason that it won't, and that being the reason that it won't dissolve in water. So we've got our polar heads that do dissolve in water. We've got this layer that is nonpolar lipids and this layer that is nonpolar lipids. So nothing's going to be able to dissolve through here and come straight into the cell unless it is also nonpolar. It has to be nonpolar to be able to diffuse across the cell membrane because it has to be nonpolar to make it through this section of lipids. Also, a lipid bilayer is just going to form spontaneously anytime a collection of phospholipids is placed in water. That's because of their affinity. Because of the polar end and the nonpolar end, we're always going to end up with this organized line of polar on the outside, nonpolar tails on the inside, and then nonpolar tails on the inside, and polar head facing the inside of the cell. The interior of that lipid bilayer is completely nonpolar. That's where our lipids are. So it will be, it will not be water soluble, and water soluble molecules, no water soluble molecules will be able to freely cross through it. Also, we find our cholesterol in the interior of this bilayer, and that's what affects the fluid nature of the membrane and provides the right amount of rigidity to the membrane. We also notice that cholesterol, when it accumulates, when we have really high levels of cholesterol, this can accumulate in the walls of blood vessels, and that can cause plaques. Those plaques could then go on to cause problems and lead to things like cardiovascular disease. Another major component of that membrane is a collection of membrane proteins. So I told you guys, some things are able to come across that phospholipid bilayer just by diffusing, specifically nonpolar things. But we have a lot of things that are polar that need to get in and out of the cell as well. And so they have to find specific, you could think of them like tunnels or channels that allow them to cross the membrane even though they don't have the right nonpolar composition to make it through the phospholipid bilayer. And the way they do that is by looking for these transmembrane proteins. So think trans like transit, we're moving, and membrane, so we're moving across the membrane through these proteins. Other proteins are also integrated into the structure of the cell membrane. One example of that is cell surface proteins, and these are going to be attached to the outer surface of the membrane and they act as markers. So basically like a name tag saying what kind of cell this is, which could provide information about where that cell should be in the grand scheme of things. Here's a picture that shows our proteins that are embedded within the phospholipid bilayer. You can see we've got our polar head, nonpolar tails, polar heads. So this will be the outside of the cell. This is the inside of the cell. And here is a protein channel that's going to allow things to pass through that could not diffuse through this layer. We also have an example of a cell identity marker. So that is shown here. This is also composed of protein, and its main goal is just to identify what kind of cell this is, which could be useful information in directing where that cell ends up. When we look at cell types, there are really two major cell types. So if we zoom out to all the different cells in the world, we've got our prokaryotic cells and our eukaryotic cells. Prokaryotic cells are going to be those that lack a nucleus and do not have an extensive system of internal membranes. We know that all bacteria and archaea have this cell type. But everything else falls under the eukaryotic cell type. These cells do have a nucleus, which is where we store our genetic information. And they will also have internal membrane-bound compartments. So any organelle within a eukaryotic cell, whether it be the nucleus or the Golgi bodies or the endoplasmic reticulum, the mitochondria, all the different organelles inside a eukaryotic cell all have their own internal membranes around them. And so that's another defining feature that separates eukaryotic cells from prokaryotic cells. And all organisms other than bacteria or archaea are going to have this cell type. So here we can look at the main differences. Eukaryotic cells will always have this nucleus to house the genetic information. As you can see, they also have all of these membrane-bound organelles with all of these different functions. When we look at prokaryotic cells, there's no specific nucleus to contain a really organized form of DNA, and there are no membrane-bound organelles. So you can see we do have this jumble in here, but that does not actually have a membrane surrounding it. Prokaryotes are going to be the simplest cellular organisms. They have a plasma membrane surrounding a cytoplasm without any interior compartments. So they do still have a plasma membrane around the outside of the cell, but they do not have any membranes on the inside of the cell. Some bacteria will also have an additional outer layer to that plasma membrane. Some are composed of cell walls with carbohydrates to give a rigid structure, and some bacteria have what we call capsules that may surround the cell wall. So they could have additional layers for protection on their exterior membrane, either being a cell wall or a capsule. The inside of prokaryotic cells has a very simple form of organization. There's not a lot to it. So we have the cytoplasm, which is uniform with little or no internal support. And when we say cytoplasm, you could think of it like the semi-fluid substance that everything's floating in. Prokaryotic cells also have ribosomes, which are the sites for protein synthesis, and these are scattered throughout the cytoplasm. And then they have what we call a nucleoid region. So they don't have a nucleus, because it would have to be membrane-bound to be a nucleus. But they do have a general area where DNA tends to be, and we call that the nucleoid region, because it is the general area where DNA is, but since it's not bound by a membrane into a specific organelle, we don't call it a nucleus. Here you can see an example of that. You can see we've got our ribosomes that are scattered throughout, which have the responsibility of making proteins. You can see the nucleoid region is just wherever the DNA hangs out. So that's the general region that the DNA tends to exist in. And then the cytoplasm is the semi-fluid substance that those ribosomes and DNA are floating within on the inside of these prokaryotic cells. You could see in some instances, instead of just having a cell membrane, we also have a cell wall, and then sometimes even further, these archaea and bacteria will have a capsule on the outer layer of that cell wall as well. These cell types also often have flagella, which is like this whip-like structure here at the end. It kind of looks like a tail. There could be one of these flagella or many, and this aids in locomotion and also feeding. So with kind of a circular whip-like motion, that is able to either propel this cell forward or to the side or in a kind of zigzag motion, or by creating kind of a helicopter-like spinning motion, they can also create a current that's going to pull food sources towards the surface of these cells. So they can basically create a current that is going to pull food to them. We also look at the pili, which are these hair-like structures around the outside of prokaryotic cells. And these can aid in attaching to substrates and also exchanging genetic information between cells. So you could almost think of them like a sticky USB cord. They're going to stick to different cells or different surfaces, and they will be able to exchange genetic information by doing that. Here's just a more close-up view of that. So do keep in mind, prokaryotic cells do not have a nucleus. They do not have membrane-bound organelles. They do have ribosomes to create proteins. They have a nucleoid region, which is where we find their DNA. They'll have a cell membrane, possibly a cell wall, possibly a capsule. Pili are used for genetic information exchange, and flagella are used to move these cells and also create currents to bring food to them. Eukaryotic cells are so much more complex, and that makes sense. We are much more complex organisms than archaea and bacteria, so it makes sense that our cells are more complex. Eukaryotic cells do have a plasma membrane, which encases the cytoplasm, which is a similarity. And we also have internal membranes that form compartments that we call organelles. I told you guys to think of organelles as the organs of the cells. The cytoplasm is semi-fluid and contains a network of protein fibers that form a scaffold called a cytoskeleton. So you could think of the plasma membrane encasing this almost jello-y substance called cytoplasm. But we have to have some kind of structure to our cells. So we get that from these protein fibers that are called, and we call that the cytoskeleton, which is like the skeleton of the cell. We also have many organelles that are immediately noticeable under the microscope. So the first of which you're going to usually see is the nucleus, because that's the really big membrane-bound compartment where our DNA is. And that's what actually gives eukaryotes their name. So in Latin, it literally translates to true nut, because the first people looking through a microscope thought, wow, it looks like these cells have a little nut inside them. What is that? And that's how we ended up with the name eukaryotic cells. When we talk about the endomembrane system, this is what gives rise to the internal membranes that are found in the cell. And each compartment can provide specific conditions that will favor a particular process. So endomembrane, think inside membrane. So inside the cell, the membrane system. Not all eukaryotic cells are alike. We notice that the cells of plants, fungi, and many protists have a cell wall beyond the plasma membrane. And also that all plants and many protists will contain organelles called chloroplasts, which are used for photosynthesis. Another unique organelle to plants is a central vacuole. This is basically for water storage. When you look through a microscope, it almost looks like the shape of a little swimming pool. And it's basically where plant cells can store excess water so that they don't just explode when they're overwatered. It's a storage mechanism. And something unique to animal cells are the centrioles. So these centrioles are like skeletal support for the animal cells. The centrioles can give rise to those protein fibers that are going to make up the cytoskeleton. Don't let this picture overwhelm you too much, because if you just repeat these definitions to yourself 5 to 10 times, they will really start to stick for you. It just takes repetition to get to remember what each organelle does. But we're going to start with number 1. Here's our plasma membrane. We already looked at a more zoomed in view of that, which looked like this. So this is our lipid bilayer and the membrane proteins that allow things to pass through that membrane. As we go on to number 2, you can see our cytoplasm. This is just the semi-fluid liquid matrix that contains the nucleus and all the other organelles. So this is what everything's floating in. Third, we have our mitochondria. If you're talking about just one, you could call it a mitochondrion, but usually people are talking about more than one, so we say mitochondria. This is the organelle in which energy is extracted from food during oxidative metabolism. So you might have heard that meme or seen that meme of, I don't know much about biology, but I know mitochondria is the powerhouse of the cell. Well, this is the little guy that we're talking about with that. And that's because any amount of energy that we're getting from any food that we've ingested is going to be achieved by the processes that happen in the mitochondria. That's where we actually get the energy from our food sources. When we talk about the cytoskeleton, this is the organelle that supports the cell shape and plays a role in cell motion as well. So you could almost think of it like the tent poles inside of a tent. So if this cell were, like a tent, and there were no tent poles, it would not be able to keep any kind of organized structure. But once we put those tent poles inside the outer casing of the tent, now we have some kind of form and stability. And that's what we're getting from our cytoskeleton. There are 3 main components, 3 different kinds of protein fibers that are acting in different ways for support. We've got our microtubules, which are going to be protein molecules in the cytoplasm, centrioles, cilia, and flagella. We also have our intermediate filaments. These are going to be intertwined protein fibers, almost braided, that provide support and strength. And then actin filaments, which are twisted protein fibers that are responsible for cell movement. So these are the cytoskeleton elements, the actin filaments, that are able to kind of almost squeeze and release in a specific rhythm that's going to allow for cell movement. When we look at number 5, that's our nucleus, which we call the command center of the cell, because this houses all of our genetic information that's controlling everything. Inside the nucleus, we have the nucleolus, which is the center of the nucleus, and that's where our ribosomes are made. So we already talked a little bit about ribosomes. We know that they're responsible for making proteins, and this is where they come from. All of the ribosomes that you see out here floating around, these little dots are ribosomes. They're all made in the nucleolus within the nucleus. The outside of our nucleus we refer to as the nuclear envelope, and that's because there is a double membrane that separates the nucleus and the cytoplasm, so that's kind of unique to the nucleus. Things that need to get through that nuclear envelope are going to come out of what we call nuclear pores. You can see them here. And they are openings embedded with proteins that regulate passage into and out of the nucleus. So they're like little channels in and out. And then you can see our ribosomes here. Like this one looks like it has just come out of this nuclear pore. They are small complexes of RNA and protein that are also the sites of protein synthesis. So that's where we are making all the proteins that we need. When we go past that, you can see we are, imagine that we've just come out of the nucleus, as some kind of genetic information that is going to code for something. And the first thing we're going to do is go through the rough endoplasmic reticulum. So, endoplasmic reticulum. We tend to shorten that and call it the ER for endoplasmic reticulum. So this is the rough ER, and you can tell it's the rough ER because it's got all these ribosomes all over it. So that can be your cue whenever you're looking at a cell or trying to label a diagram. If it's this part around the nucleus that has ribosomes all over it, that's going to be the rough ER. And this is also an area of major protein synthesis. That's why we have so many ribosomes concentrated in this area, because a lot of proteins are going to be made in the rough ER. Yes, it's possible that proteins will still be made over here, or here, or here on these ribosomes, but the majority of proteins that we're going to end up with are going to be processed through this rough ER. That's not all of the ER, though, because we also have the smooth endoplasmic reticulum, the smooth ER. And you can tell it's the smooth ER because there's not any rough ribosome bumps on the surface of it. This is where we're manufacturing our carbohydrates and lipids. So, in the rough ER, we're making proteins. In the smooth ER, we're making the carbohydrates and lipids that we need. We're also going to want to keep these two separate. So, you can see for number 7, we've got lysosomes, which is like this little sphere here with enzymes inside it. And this is also number 7, peroxisomes, which is a similar vesicle or small sphere with enzymes inside it. The difference is these lysosomes are used to digest and break down worn out parts of the cell that we're going to reuse. So for instance, if this mitochondria had just broken down and was no longer functional, the lysosomes would attach to it, the enzymes would break down the cellular components so that we could use those pieces to create new things within the cell. Lysosomes are also important for just digesting food. So we use lysosomes to break down the food that we have ingested as well on a cellular level. Peroxisomes function the exact same way. They're going to attach to things, release the enzymes that will surround whatever needs to be broken down so that we can get rid of them. But these are targeting potentially harmful molecules. And so my little trick for remembering which one's which is to think of hydrogen peroxide as a cleaning agent, because lysosomes are made to break down worn out parts of the cell and food, but peroxisomes, like hydrogen peroxide, are used to clean and get rid of harmful molecules. So maybe that trick will help you keep them separate, but they look basically the same, they function the same. If it's a lysosome, it's breaking down worn out parts of the cell or food. If it's a peroxisome, it's breaking down harmful molecules. Let's see. We also have our centrioles here, and this is where our cytoskeleton is coming from. So this is basically the assembly center for our microtubules. And the centrioles always show up in pairs, and they create our microtubules in these 3 system chains. So they basically give rise to our cytoskeleton, which you can also see represented here. So there's our different branching kinds of microtubules and protein filaments that are providing structure to our cell. You can see this secretory vesicle here, which is a vesicle that is fusing with the plasma membrane, releasing materials to be secreted from the cell. So that's a pretty common thing of coming and going from the cell. We're going to basically have a membrane that folds on itself either to go in or that folds outward and creates a vesicle like this one to go out. When we're looking at plant cells, you'll notice a few different things. So first of all, we still have our plasma membrane. It's right here. Let's see, number 2, we still have cytoplasm. So that's the semi-fluid stuff that everything else is floating in. We've got mitochondria. We still need those in plant cells to get energy. And here is our cytoskeleton. once again made up of microtubules, intermediate filaments, and actin filaments. So that's the same as our animal cells. 5, we have our nucleus. It's got the nucleolus inside it to make ribosomes. We've got our nuclear envelope surrounding the nucleus, a nuclear pore for things to come out or go into the nucleus. Once again, we have our rough endoplasmic reticulum and our smooth endoplasmic reticulum. And then we have our Golgi complex. Sorry, I may have forgotten to point this one out on the previous slide, but it's the same for both of these, plant and animal cells. The Golgi complex you could think of as packaging and distribution. So whether it's a protein that needs to be packaged to go somewhere, or a carbohydrate or lipid coming from the smooth ER that needs to be packaged to go somewhere, it will next come to the Golgi complex, and that's how it ends up wherever it needs to go. So think of Golgi complex like UPS. It's the delivery system. It's organizing things and taking them where they need to go. Here again we have our peroxisomes. So you can see the peroxisome here. Over here we have our cell wall. So that is something very unique to plant cells. We have a cell wall to provide that extra structure so these cells don't burst. Some are, and then we have an adjacent cell wall as well. In plants, adjacent cells are glued together by a sticky substance between their cell walls. We call that the middle lamella. Let's see, we have our chloroplasts. So these are also unique to plant cells and protists. So these are the organelles that contain thylakoids, and they are the site for photosynthesis. So the actual process of getting energy from the sun is happening in these chloroplasts. And another unique feature to plant cells is the central vacuole. This is the one I mentioned before that looks kind of like a swimming pool. In plants, it's a storage compartment for water, sugars, ions, and also different pigments. So it's basically like a big holding tank for all of the things that those cells are going to need. As we mentioned, the nucleus is the command and control center of the cell. That's where we're storing all of our hereditary information. And the surface of that nucleus is bound by a double membrane, which we call the nuclear envelope. Groups of proteins will form openings called nuclear pores that allow proteins and RNA to pass in and out of the nucleus. The DNA of eukaryotes is going to be packaged into segments and associated with proteins. That's just our way of organizing that DNA. This complex is called a chromosome, and the proteins enable the DNA to be wound very tightly and condense during cell division. So you can imagine if we're trying to split a cell in half and end up with the right amount of DNA in the two daughter cells that we've created, we need to have an organization to that DNA. Otherwise, one of the daughter cells could end up with 3/4 of the DNA, while the other daughter cell gets 1/4 of the DNA. So we need an even split, and having organized DNA is what makes that possible. So whenever the cell is not dividing, the chromosomes will exist as thread-like strands called chromatin. So you can see that here. Here's chromatin. Just long strands of DNA. But once it is time for the cell to divide, that's whenever the chromatin will condense into this chromosome form. Also, protein synthesis is going to occur when the DNA is in that chromatin form as well. Once we shift into cell division, the chromatin will be in this chromosome form, and that's going to allow for the correct distribution of DNA to the daughter cells. The cell builds proteins on structures called ribosomes, so we mentioned those. Those were those little dots that came out of the nuclear pores here. And ribosomes consist of ribosomal RNA and also several different kinds of proteins. Those ribosomes are assembled in a region of the nucleus called the nucleolus, here at the center, and then they come out through the nuclear pores. So the nucleus holds the chromosomes, or the DNA. The nucleolus is the place where the ribosomes are made within the nucleus. You can see that again here. We've got our nucleus with the nucleolus at the center. Ribosomes will be made here. They will leave through the nuclear pores. Remember that this is called the nuclear envelope. The endoplasmic reticulum is that extensive system of internal membranes that results in all of our proteins, carbohydrates, and lipids. Some of the membranes form channels and interconnections, and other portions will become isolated spaces enclosed by membranes, which basically means they'll become vesicles, like those lysosomes that we saw. Basically, if this little section broke off and was a floating sphere, that would be a vesicle. And that's how a lot of carbohydrates, proteins, and lipids are going to be transported from the ER to the Golgi bodies. So we've got our rough and our smooth ER. You can tell, again, as I said, rough ER because it is covered in those ribosomes, and that makes it look really pebbly. The portion of the ER that aids in manufacture of carbohydrates and lipids is the smooth ER. It looks smooth because it doesn't have ribosomes on top of it. So this is a good slide to help you remember if you want to screenshot or just take a picture of this one. Rough ER, let's make proteins. Also, you could think rough like ribosomes, R and R. You know, if the rough ER has ribosomes and ribosomes make proteins, then that must be the part of the ER involved in protein synthesis. When we look at the smooth ER, this is where we're getting our carbohydrates and lipids. So there are no ribosomes present on the surface because we're not trying to make proteins, we're trying to make carbs and lipids. After the rough and smooth ER, you'll see the Golgi complex here, where we go to for packaging and distribution. The Golgi bodies are also known as the Golgi complex, and the newly made molecules are going to end up here. That's so they could go into the flattened stacks of membranes that are scattered through the cytoplasm and be collected, packaged, and distributed to different areas of the cell. When we're talking about the Golgi bodies, like all of the Golgi bodies within a cell, we're talking about the Golgi complex. Here you can see some of those transport vesicles. We can end up with vesicles that come off of the ER, and also vesicles that will leave the Golgi complex as well. So basically, whatever we have made that needs to be transported is going to be surrounded in this outer membrane and then enclosed and kind of pinched off and then free to be transported wherever it needs to go. We call those secretory vesicles. So here you can see that process happening. We go from the rough ER into through this vesicle, transport vesicle from the ER, we will come to the Golgi complex. We can leave as lysosomes, which are going to be useful for breaking down worn out parts of the cell or different kinds of food. These vesicles could also leave and end up going outside of the cell, just like what we see happening here. So this vesicle is coming and attaching to this vesicle to break something down. This vesicle is going to cozy up to the edge of this plasma membrane where the outside of the cell is, and then basically fuse with that lipid bilayer so that the inside of the contents can be expelled out of the cell. So that's how we're getting stuff out of the cell. The Golgi complex also gives rise to lysosomes. These membrane-bound structures will contain those enzymes that the cell uses to break down macromolecules. As I said, we're breaking down worn-out cell parts and food particles. Particles that the cell has ingested are also going to need to be digested. So here you can see a figure of the lysosomes at work. Here we have a lysosome attaching to an old or damaged organelle. Those enzymes come in to break down the pieces of that organelle. And once those pieces are broken down, they could be expelled from the cell, or they could be recycled to be used again. In another case, this lysosome could come up to a food vesicle that has entered the cell through the plasma membrane. So this part of the plasma membrane closed around the food vesicles, around the food, to create a food vesicle that could then merge with this lysosome so that we could use our enzymes to break down that material and get energy from it. When we're talking about peroxisomes, they're still vesicles and they have that same basic motion and function, but they're actually working to detoxify different potentially harmful molecules. And they do this using molecular oxygen to remove hydrogen atoms from specific molecules, which is basically going to break them down on a molecular level. Another interesting thing is peroxisomes in plant seeds can also work to convert fats into carbohydrates, which are needed. When we talk about organelles that contain DNA, this could be kind of confusing because we just mentioned how all of the genetic information is housed in the nucleus. But this is different because these are two organelles that have a different kind of DNA. It is within the chloroplast and the mitochondria. They seem to have been derived from ancient bacteria that were taken up by eukaryotic cells. So it's not the same as our long strands of chromatin and chromosomes. They actually have these rings of circular DNA. Both of these organelles have that in common, this circular DNA. Mitochondria are known as the powerhouse of the cell because they are the sites for chemical reactions, which we call oxidative metabolism, which basically just means that's where we're turning our sugar into energy that we can use. That's why it's called the powerhouse of the cell. The organelle is surrounded by two membranes as well. You can see the outer membrane here and the inner membrane here. When we're looking at our chloroplasts, these are the sites of photosynthesis, and this organelle is also surrounded by two individual membranes and also has that circular DNA present inside it. Both mitochondria and chloroplasts possess their own molecule of circular DNA, and neither of them can be grown outside of the cell. They are totally dependent on the cell within which they occur. So just because they have their own circular DNA, that does not mean they're a cell within a cell, because they cannot function without everything else inside the cell. And this is what led to the theory of endosymbiosis. This theory states that some organelles had to have evolved from a symbiosis in which one cell of a prokaryotic species was engulfed by and lived inside of a cell of another species of prokaryote. And that was a precursor to eukaryotic cells, or eukaryotes. The idea is that the engulfed species provided their hosts with advantages because of those special metabolic activities, like oxidative metabolism in mitochondria and photosynthesis in chloroplasts. The modern organelles of mitochondria and chloroplasts are also believed to be found in the eukaryotic descendants of these endosymbiotic prokaryotes. And this is all we're talking about. The idea is we had a really basic prokaryotic cell that engulfed another prokaryotic cell, and through that benefit that was achieved from their existence together, we, through evolution, ended up merging these cell types into eukaryotic cells that could be more complex organisms. In addition to the double membranes and circular DNA found in mitochondria and chloroplasts, there's also a lot of evidence supporting the endosymbiotic theory. The first is that mitochondria are about the same size as modern bacteria. Also, the cristae, those folds inside the mitochondria, resemble folded membranes that exist in modern bacteria. Mitochondrial ribosomes are similar to modern bacterial ribosomes in both size and structure. Also, mitochondria divide by fission, just like modern bacteria, which we haven't really got to yet, but you could just keep in mind that mitochondria inside eukaryotic cells uses the same division mechanism to create more mitochondria that bacteria uses to create more bacteria. And that's called fission. When we look at the cytoskeleton, it is comprised of an internal framework of protein fibers that anchor organelles to fixed locations, that also support the shape of the cell, and that will help organize ribosomes and enzymes needed for synthesis activities. So the cytoskeleton is dynamic, and its components are continually being rearranged. So here are the main three components separated in a way that you'll never actually see them. But it does make it easier to visualize the structure of the intermediate filaments compared to the microfilaments and the microtubules. That's why I included this image. The three different types of protein fibers that make up the cytoskeleton are the microfilaments, which are those long, slender microfilaments made up of protein actin. So you can see those here. Those are our actin filaments. We also have microtubules, which are the hollow tubes made up of the protein tubulin. So here's one of those hollow tubes that go throughout. And then we also have our intermediate filaments, which are like thick ropes of intertwined proteins, providing a lot of structure as well. Here you can see another representation of those three forms that make up the cytoskeleton. And we have our centrioles. So these are the complex structures that actually assemble those microtubules in the cytoskeleton. And they are in animal cells and also the cells of most protists. They occur in pairs. They are usually found near the nuclear envelope of the nucleus. And they are themselves composed of microtubules as well. So microtubules making microtubules. But the source is the centriole. Here's what they look like. And we are usually forming those microtubules in these kind of triplet formations that you can see here. So this is another feature that's unique to animal cells and some protists. Plant cells will not have these centrioles. When we're talking about vacuoles, there are two main types of vacuoles that we will find in cells. The first we already mentioned, the central vacuole, which stores water and other substances in plants. That's the one that looked like a big swimming pool. But there are also other cells that have a contractile vacuole, like a lot of protists. And this vacuole will be found near the cell surface, and it will accumulate excess water from inside the cell and then actually pump it out. So here you can see a central vacuole, and here's what it looks like under a compound light microscope. There we can see the chloroplast under the compound light microscope. And then here's what a contractile vacuole would look like. But you kind of need to really see what's happening here. So here we can see it filling with water and expressing it. Here it is. And once it squeezes, it empties that water out. So again, it accumulates all of that excess water and then pushes it out. That is a contractile vacuole. You could think of it almost like a water balloon filling with water and then being squeezed out of the cell. So that's our example of a contractile vacuole on a paramecium cell. And when we talk about cellular motion, it's usually associated with the movement of those actin microfilaments, like the cytoskeleton microtubules. Some cells are able to crawl by coordinating the rearrangement of those actin filaments, kind of like an inchworm motion. Also, some cells are able to swim by coordinating the beating of microtubules that are grouped together to form either flagella or cilia. When we're looking at the differences between cilia and flagella, they are so similar. They're both kind of hair-like structures that extend from the surface of the cell and are involved in movement. But usually cilia are short and numerous, so there are lots of them. They're hair-like projections that will move cells or fluids using a coordinated wave-like motion. So you could kind of imagine the way that seaweed waves in the water. That's what the cilia motion would look like. And the flagella are those long whip-like appendages that are going to propel cells with a rotating movement. So more like the blades of a helicopter, creating that kind of movement forward or creating a current to pull food towards this cell. The endomembrane system is also useful for moving materials short distances in the cell. And for long distances, lysosomes will move along those microtubular tracks. So we've got those protein filaments that lysosomes, those small vesicles we saw, can attach to and follow to find where they're going. And that actually makes it more efficient for them to find where they're going, because instead of just trying to, you can imagine it like instead of trying to go straight through thick woods with a bunch of brush and you don't know where you're going or which direction to go, instead if you were just walking alongside railroad tracks to your destination, that's a clear-cut path that's going to make it more efficient for you to get where you're going. And that's what it's like when these lysosomes follow those microtubular tracks. They will move to food vacuoles to break down different kinds of food and get energy from them. And mitochondria will also travel down along long nerve axons whenever they need to travel a different distance. So, long-distance intracellular transport, meaning getting a long distance across the cell, that is going to require the following components. We're going to have a vesicle or an organelle, which is pretty much the material to be transported. And then a motor molecule, which will usually be either dynein or kinesin. And then we'll have a connector molecule that connects the vesicle to the motor molecule. And then those microtubule tracks, and that's what the vesicle is going to move along. So it looks kind of like this. Here's our microtubule, which you could think of like the train tracks. This is, in this instance, it's dynein, but this is our motor molecule. So you could think of this almost like the train itself that is carrying something. Here are our connector proteins that are connecting to the vesicle, which is like the cargo that's being carried. So these are descriptions that just kind of further flesh out all of the organelles we've just talked about. If you want to screenshot this or pause and take note of this, you're welcome to. I included it because it is useful, but I've just told you about these organelles, so I'm going to leave that to your screenshotting. Here's another slide. Same thing. If you'd like to screenshot this and come back to it or take notes, you're most welcome. And when we talk about plants, fungi, and many protist cells that have cell walls on the outside of their cells, it's important to remember that animal cells do not, and some protist cells do not. So I told you guys how protists are kind of the weird kingdom that have all of these different features. I told you some are unicellular, some are multicellular. Some do have cell walls like plants, and some don't have cell walls like plants. So there's just a lot of variety in protists, but it is important to remember that animal cells are not going to have cell walls. That is for plant cells and some rare protists. Those plant cell walls are going to include the primary walls, which are laid first, and then they're going to have what we call the middle lamella, which is what sticks those adjacent cell walls together, and then the secondary walls, which are laid inside the primary walls. So that looks like this. Here we've got our secondary wall on the innermost part of the cell. Our primary wall faces the outside of that cell. And then the area between two cells, so here's primary wall and primary wall. The area between those two cells is what we call the middle lamella. Animal cells can secrete an elaborate mixture of glycoproteins, and they do this to form the extracellular matrix. So this is the matrix outside of the cell that forms a protective layer over the outside of the cell. It does not provide support like a cell wall, though. It looks something like this, so it's very similar to the wild kind of protein network inside the cell. It's also going in every different direction on the outside of the cell whenever it's these different parts of the extracellular matrix. when we talk about movement of materials across the cell, there are basically 3 ways to cross that plasma membrane, the cell membrane, the outside of the cell. It's either going to happen through diffusion, membrane folding, or transport through membrane proteins. When we talk about diffusion, this is when molecules move in a random fashion, but there is a tendency to produce uniform mixtures. So the net movement of molecules from an area of higher concentration to an area of lower concentration. That's what we're talking about with diffusion. And these molecules will diffuse down a concentration gradient from higher to lower concentrations. Diffusion will end when equilibrium is reached, which means there's basically a balanced amount of that substance in that specific area. So you can imagine if we're in the classroom and I spray some really strong perfume at the front of the room. That perfume is going to diffuse throughout the room until the molecules are spread evenly throughout the room. And there's no energy that has to be exerted to make that happen. It's just happening because of the concentration gradient from high concentration of basically a lot of perfume in one area to lower concentration, the areas of the room that don't have any perfume in them yet. So that's one way to come across the cell membrane. That is diffusion. And here's another example where you can see that happening in water. Diffusion is what happens when water breaks up into, when sugar breaks up into water to form this solute. So here you can see we've added a lump of sugar. We've got a really high concentration of sugar molecules here now. Those are going to diffuse throughout the water until we have an equilibrium, which means we've got equal distance surrounding each of these particles of sugar. When we talk about selective permeability, that's just describing how it basically is what allows cells to control what comes and goes through the plasma membrane. So, permeability, that's the ability to come in or go out, to come through a material or a substance. And selective, meaning not just anything can come through or go out, it's selective, only certain things can. So, like I mentioned before, only non-polar molecules are going to be able to pass through our phospholipid bilayer. Those are things like O2, CO2, N2, also small fats and lipids, non-polar things that are going to be able to diffuse through this plasma membrane. Any kind of polar molecule that needs to come into the cell is going to have to find a way in, because anything that's an ion, meaning it has a charge, whether it's positive or negative, or anything that has polarity, like this water molecule, is not going to be able to pass straight through this non-polar portion of the phospholipid bilayer. So that's why we're going to have to find another way in for those guys. Only certain substances will undergo diffusion across that plasma membrane. Like we just mentioned, molecules like oxygen, carbon dioxide, and non-polar lipids will be able to diffuse, but any kind of ion or polar molecule will not. The concentration gradient is the number of molecules present per unit volume on one side of a membrane compared to the other side. So we know that molecules will move to areas of a lower concentration without any kind of energy expenditure. Proteins are going to act as carriers that can bind only to specific molecules and transport them. Transport will be limited by the availability of carriers, and when all of the carriers are in use, that's when the transport is considered saturated. So here you can see in step 1 with facilitated diffusion, our particle molecules can bind to special protein carriers in the plasma membrane. The protein carrier will help or facilitate the diffusion process and does not require any energy. And the molecule will then be released on the far side of the membrane. The protein carriers transport only certain molecules across the membrane, but they will take them in either direction, so they could come into the cell or go out of the cell. But it's important to remember that no energy is required for facilitated diffusion. Even though we are using this channel, we're still diffusing, so no energy is required. It's just all based on the concentration gradient and the affinity to this channel. When we talk about osmosis, this is when water moves down its concentration gradient into or out of a cell, and the movement of water is dependent on the concentration of other substances in the solution or in the cell. The greater the amount of solutes that are dissolved in a solution, the lesser the amount of water molecules that are going to be free to move. So here's a great example of osmosis. Osmosis is going to always involve a semi-permeable membrane like this, and you can see diffusion causes water molecules to distribute themselves equally on both sides of this semi-permeable membrane when all we have in this beaker is water molecules. So we have an even distribution. But once we add a solute, something that's going to mix into the water, to one side, that's going to change the concentration gradient. So the addition of the solute molecules that cannot cross the membrane is going to reduce the number of free water molecules on that side as they bind to the solute. So we have water binding to this solute, and that's going to mean that diffusion will cause free water molecules to move from the side where their concentration is higher, over here, to the side where that we have our solute, where the concentration of water molecules is lower. And that's how we end up with this imbalance in our levels of water here, because we're trying to reach an equilibrium with concentration, but with this solute, it's going to require more water on this side to reach that equilibrium. Also, as we talk about this process, we have different terms for defining the concentrations. So, if the osmotic concentration of two solutions are equal, we call that an isotonic solution. So, isotonic means equal. If two solutions have unequal osmotic concentration, the solution with the higher solute concentration, so solute is whatever you added to your water, that side is going to be called hypertonic. And the solution with the lower solute concentration, like the side that had mostly water, is going to be called hypotonic. You could think of when you're hyperactive, you have a lot of energy. When you're hypertonic, you have a lot of solute. When you're hypotonic, you have less solute in an uneven distribution. Movement of water by osmosis into a cell is also going to cause pressure, which we call osmotic pressure. And enough pressure may cause a cell to swell and burst. But osmotic pressure explains why so many cell types are reinforced by cell walls, because if they weren't, that osmotic pressure would cause them to burst. When we talk about hydrostatic pressure, that is what we term the pressure of the cytoplasm when it's pushing out against the cell membrane. Hydrostatic pressure. Here you can see what our cells look like in a hypertonic solution. So that means more solute than water. And then here we have what our cells look like in an isotonic solution, where we have an equal balance of water inside and outside of the cell. That's isotonic. And then here you can see our cells in a hypotonic solution, meaning that we have more water inside the cell relative to the amount of solute outside of the cell. And then here you can see what that looks like under a microscope. Our shriveled cells, normal cells, and hypotonic cells. And then with plant cells, here's the example of hypertonic, meaning more solute than water, isotonic, meaning balanced, and hypotonic, meaning more water inside the cell than solute. Organisms have developed lots of solutions to maintain osmotic balance, and extrusion is what happens when we're using those contractile vacuoles to eliminate excess water. So like that video that we saw of the paramecium, which is one of our protists, that's an example of extrusion, where we are contracting that vacuole to eliminate excess water. Also, isosmotic solutions in the cells can prevent net flow of water in or out of the cells. So just basically by balancing those solutions, that's going to prevent that concentration gradient that's going to push water in or out of the cells. And then also turgor pressure, which is created by plants because their cells are hypertonic to the environment, causing water and pushing the cell membrane against the cell wall. When we talk about the passage into and out of the cells, we have to have ways to do that for bulky substances, and that's usually through vesicles, and we call that either endocytosis or exocytosis. We call it endocytosis when we're going into the cell, and exocytosis when we're going out of the cell. Here's what that looks like. So here's endocytosis, meaning something's going into the cell. You can see something cozies up to the plasma membrane. That plasma membrane starts to engulf that substance until it encloses it completely. The membrane separates, and we now have a vesicle inside the cell. The same thing is happening here with a liquid. And we have two different names for endocytosis based on bringing in solid matter or liquid matter. If we're bringing in solid, we call it phagocytosis. If we're bringing in a liquid, we call it pinocytosis. And if you're trying to keep those two names for endocytosis separate, I don't really have a great trick, but the way that I remembered it was thinking of Pinot Noir, which is a type of wine, and that is a liquid. So if you could think of Pinot, like pinocytosis, bringing a liquid into the cell, and then you would know the other one, phagocytosis, is when you're bringing in solid particulate matter. Exocytosis is the same process, but going from the inside to the outside. So once again, we've got a vesicle. Those membranes are going to fuse together as they meet. That's going to create this opening so that the contents from within the cell can be expelled out of the cell. We also have something called receptor-mediated endocytosis, and this is a more particular version of bringing something into the cell. And this is for molecules to be transported into the cell that are going to first have to bind to specific receptors in order to make that happen. So here's the target molecules. They're going to have to find these specific receptors. Once the correct arrangement of them has paired to those receptors, that's going to initiate the folding of that membrane so that we end up with our coated vesicle that has now been brought into the cell. But it's still endocytosis because we're still bringing things into the cell. When we talk about active transport, this is when we're utilizing those protein channels that open only when energy is supplied. So we haven't talked about any forms of active transport yet, but energy in this case is going to be used to pump substances against or up their concentration gradients. So basically, like going upstream, we're fighting the current. This allows cells to maintain high or low concentration of certain molecules. And you can recall that diffusion always ends in equilibrium. So, diffusion ends in equilibrium, but we may still need to move things regardless of that, which means we're going to have to fight the concentration gradient. And most of the active transport that happens in cells is going to be carried out by the sodium-potassium pump. So this is the number one active transport for our cell function, the sodium-potassium pump. With this pump, we are using energy in the form of ATP to pump 3 sodium ions out of the cell and pump 2 potassium ions into the cell. Almost a third of the energy expended by the body's cells is given over specifically for driving these pumps. So it's an extremely important part of our process. And here's what it looks like. You can see we've got our protein channel here that is part of our active transport. Right now it's open to the inside of the cell. That gives these sodium ions, our Na+, a chance to attach to their specific spots in that protein channel. Once they do, that's also going to allow our adenosine triphosphate, this energy molecule, to attach to our protein channel. Once that happens, we can cleave that third phosphate bond, which is going to provide a burst of energy and allow the perfect conditions for this protein channel to change shape and then open to the outside of the cell. Once it's open to the outside of the cell, we've got the perfect conditions for our sodium ions to detach and our potassium ions to attach and come into this protein channel. Once they do, that's going to have the perfect affinity for this channel to then open to the inside of the cell. We're going to release that last phosphate that is attached, that came from our adenosine triphosphate, and now we've brought in 2 potassium ions. And this just keeps going. So now you can see it's open back to the original configuration. We're going to end up with 3 more sodium ions, and we're going to end up with another adenosine triphosphate here. That phosphate bond will break. This will open to the outside, sodium out, potassium in, and so on. So that is a process that is constantly happening. Sodium out, potassium in, sodium out, potassium in, over and over and over again. And this is a great example of active transport. So the result of that sodium-potassium pump is to generate a concentration gradient with more sodium ions outside of the cell than inside. So basically, without this pump, we would just have a constant flow of sodium ions into our cells based on the concentration gradient. And so this is providing a way for us to find balance where we have more sodium ions outside of the cell. Cells exploit this gradient in key ways. 2 of the main ways is the conduction of signals along nerve cells. That is only made possible by these sodium-potassium pumps. And also the transportation of important molecules into the cell that need to go against their concentration gradient to get there. The cell membrane has many facilitated diffusion channels for sodium ions, but it is only going to be transported if it's partnered with another substance, like potassium. This is a great example of what we call coupled transport. And so the only way in this case that sodium is going to get into the cell is if it's coupled with a substance. And in this case, that's potassium. That's what allows it to go against the concentration gradient. Coupled transport is a common way for cells to accumulate sugars and also amino acids. If you're trying to remember, if you're trying to think of a way to remember coupled transport and what it means, you could imagine it's like a bridge that you have to be holding hands with somebody to be able to cross. Sodium ions cannot just leave the cell. Potassium ions cannot just come into the cell without this coupled back and forth. So it's coupled transport, meaning we have to have 2 molecules working together. Here you can see the processes we've gone over for coming into and getting out of the cell. Here are our passive processes. And when we say passive, that means no energy has to be supplied to make them happen. The examples we gave were diffusion, also just open protein channels that allow movement through the channels into the cell or out of the cell. Facilitated diffusion, where we have a protein carrier that's going to allow diffusion through a cell membrane, and also osmosis. So all of these are passive processes that do not require energy expenditure. And then here we have our active processes. So the folding of the membrane to bring things in and out, whether that is coming in with endocytosis or out with exocytosis, whether or not it is using these receptors for receptor-mediated endocytosis, these are still going to require energy. So that's why they're active processes. Also, we've got this example of active transport because this is our sodium-potassium pump, which is requiring energy expenditure to change shape from the inside to the outside. And then here is an example of coupled transport, which is requiring more than one molecule to come through to be able to go against or to go into or out of a cell. And that's it for Chapter 4. So, thanks for bearing with Chapter 4. I know it's a lot of information. If you study it a few times, it will really help all the information stick. So, find a friend to study with if you can. Watch this multiple times if that helps. You can pause and take notes. Whatever you need to do. If you have any questions, feel free to email me. I'll be happy to help. Thanks.